Today, we are going on a kind of genetic treasure hunt. We're diving deep, deep inside our own DNA to uncover a mystery that's been hiding in plain sight for, get this, over 800 million years. We're talking about a genetic stowaway with a really fascinating story to tell. So let's just jump right in with a wild thought experiment. What if one of the most fundamental genes for, building you, wasn't off on its own, but was actually tucked away, completely hidden inside an entirely different gene? I know, it sounds like something out of science fiction, but as we're about to see, this is a real-life genetic puzzle.
All right, let's start with clue number 1 in our little investigation, this truly bizarre genetic setup. Seriously, it's like finding a secret room in a house you've lived in your whole life.
Okay, so here are the two main characters in our story. First, you've got the HTR2B gene. This thing codes for a super important serotonin receptor. Then, you have the PSMD1 gene, which is part of the cell's protein recycling crew. Now here's the bombshell. HTR2B is completely nested inside a non-coding part of PSMD1, a section called an intron. It is literally a gene inside of another gene.
And listen, this isn't some new quirky mutation that just popped up. This nested arrangement is ancient. I mean, really ancient. We find it in us, in humans. We find it in zebrafish. We even find it in jawless fish like the lamprey. And we can trace it all the way back to pre-vertebrates like the sea squirt, Siona. We're talking about a genetic partnership that has been carefully preserved for hundreds of millions of years.
Okay, so if these two genes have been evolutionary travel buddies for that long, you would expect their family histories to line up, right? It just makes sense. So let's check the evidence. Let's look at their evolutionary trees.
First up, the family tree for the serotonin receptor genes. You can see how the different family members, the 5-HT2A, 2C, and our gene of interest, 2B, are all related. Now, pay close attention to how HTR2B branches off. It's got its own unique evolutionary path.
And next up, here's the family tree for its host gene, PSMD1. As you can see, it also has its own long and storied evolutionary history. And here it is. This is the aha moment. When you put the evolutionary tree for just the HTR2B gene right next to the tree for its host, PSMD1, they're a near-perfect match. It's like looking in a mirror. This is our smoking gun. This is definitive proof that these two have been evolving together, in lockstep, for eons.
So how in the world did this even happen? And why is this one pair so special, while all its relatives branched off and did their own thing? Well, the answer goes back to these huge, ancient events called whole genome duplications, basically, a time when an organism's entire genetic code gets copied.
Let's walk through how this all went down. So we start with an ancestor like Siona, and it's got one copy of our nested pair. Then, bam, a massive genome duplication happens. Now, logically, you'd think you'd end up with 2 copies of everything. But something really weird happens. One copy of our nested pair just gets lost. Then, it happens again and again. With every one of these duplication events, the other serotonin receptor genes multiply, but our little HTR2B/PSMD1 pair stubbornly stays as a single copy. So, why? This was not random chance. This wasn't just some accident. As the researchers put it, there's a strong selective pressure to keep only one copy. This means evolution is actively, purposefully making sure that this pair stays unique. And that tells us it must be doing something incredibly important.
So what is that super important job? What's the motive behind this strange, conserved relationship? Well, it turns out this genetic stowaway is holding the blueprint for some of the most fundamental features of a vertebrate body.
Just take a look at these incredible embryo images from the study. The 5-HT2B receptor is super active in what are called neural crest cells. Think of these as the master builder cells that travel all over the embryo, and they're responsible for forming the bones of your face, your jaw, and big parts of your nervous system. Essentially, this gene helps build the new head that was a key innovation for all vertebrates.
And its role is also absolutely critical for the heart. I mean, look at these images. They show the heart of a mouse embryo. On the left, you see a healthy developing heart. But on the right, that's an embryo where the HTR2B gene has been shut down. The heart is severely underdeveloped. The muscle walls are thin. The whole structure is just compromised. This one gene is absolutely required to build a functioning heart.
And the gene's job doesn't stop there. Nope. It's also essential for creating our entire blood system. This slide shows an embryo without a functional HTR2B gene. Just look at how pale it is compared to the healthy one on the left. That paleness, it's a direct result of problems with making blood cells, a process called hemopoiesis.
And you know, these effects aren't temporary. They are permanent. This table here summarizes data from adult mice that are missing the gene, and it shows lasting changes in key cell types in their blood and bone marrow. So this gene's role isn't just a temporary gig during development. It literally establishes the foundation for life.
So now we have all the clues, right? We have the nested structure, the matching family trees, the weird resistance to being copied, and all these vital functions. Let's put all the pieces together and finally solve this thing. So what did we find? A unique gene within a gene structure that's been conserved for over 800 million years. We found evolutionary family trees that are a perfect match. We found this stubborn refusal to be duplicated even when all its relatives were multiplying, and we found that it's essential for building the absolute core components of a vertebrate: the face, the heart, and the blood system. All of this evidence points to one powerful conclusion, what the researchers are calling the ancestor hypothesis. The theory is that this nested gene, HTR2B, isn't just another member of the family. It's the original. It's the common ancestor of the entire 5-HT2 receptor family. And its ancient partnership with PSMD1 is so critical that evolution has locked it in place for nearly 1 billion years.
I mean, it is a stunning piece of evolutionary detective work, and it leaves us with this one last, fascinating question to think about. What other ancient secrets, what other genetic stowaways are hiding in plain sight right now, inside our own DNA, just waiting for us to find them?